So when I was asked to introduce Dr. Thomas Insel, I jumped at the opportunity because I'd heard so many good things about him, but I hadn't met him. And even though I've had funding from the National Institute of Mental Health, NIMH, for the past 13 years, I'm not a psychiatrist, I'm a statistician. And the request for an introduction came less than 48 hours ago on Sunday afternoon. So I tried to get some advice by emailing David Lewis, the chair of the University of Pittsburgh's Department of Psychiatry and director of its Western Psychiatric Institute and Clinic. David instantly emailed me back, and if you know David, I guess that's not a surprise. He told me he was on the beach with his family. But he had recently spoken to Tom, and he knew Tom was looking forward to the visit. And then David referred me to that most authoritative source for biographical information, the World Wide Web. So here's my summary of Google search results, and I just hope it's halfway accurate. First, according to the New York Times, Dr. Insel entered college, actually a combined BA-MD program at Boston University at the age of 15. Then he took a break 2 years later at the age of 17 to backpack all over the world. This was the late 1960s. But he made his way back to school, and after graduation did an internship and residency in psychiatry and went to work at NIMH doing clinical research. In 1994, he moved and became head of the Yerkes Primate Center at Emory University, and again, according to the New York Times, he purchased his first necktie because his new job, among other things, required him to play a public role. He must have played that role well, because in 2002 he became director of NIMH and is now the longest-serving director since its founding director retired almost 50 years ago. From perusing his publications, and there's more than 200 of them, it's apparent that Dr. Insel's early clinical studies were centered on obsessive-compulsive disorder, OCD, and he was lead author on a 1983 double-blind, randomized, crossover design study, so part of the first wave of statistically rigorous studies in psychiatry, which found that a serotonin reuptake inhibitor was effective in reducing symptoms associated with OCD. So I gather that this was one of several studies that directed attention to selective serotonin reuptake inhibitors, or SSRIs. This seems to have led him to benzodiazepine receptors, apparently because of the role of anxiety in OCD, and the relationship of benzodiazepine receptors to anxiety. Then he wrote about animal models of anxiety, which included separating infant animals from their mothers, and perhaps this pushed him toward the study of parental attachment to their offspring. In any case, whatever his initial motivation might have been, Dr. Insel spent a chunk of his career on the neurobiology of attachment, where his studies of the role of oxytocin and vasopressin in mating and parenting behavior became very well known, including the remarkable demonstration that injection of a vasopressin antagonist changes male prairie voles from being monogamous to promiscuous. Surely one of the quintessential results on the molecular basis of bad behavior. Dr. Insel also wrote a book called The Neurobiology of Parental Behavior. For his many research contributions, he's received numerous honors and awards, including election to the National Academy's Institute of Medicine. One more thing. Dr. Insel writes a blog, and it turns out its content is interesting. This at first surprised me, because usually when I listen to or read comments from those high up in the NIH structure, I find the remarks fairly bland, not because those people aren't full of good ideas, but because the political aspect of their position seems to make them very cautious. It's obviously a difficult job to navigate simultaneously the science and the politics of their subject, but Dr. Insel does a terrific job. And by the way, in one of the entries, there's a quotable statement that neuroscience research is the key to our being able to preempt the development of serious mental illness. In leading the NIMH, Dr. Insel has been a strong advocate of the need to discover the biological basis of mental health, including being a leading proponent of the Research Domain Criteria, or RDoC, a new framework for classifying mental disorders based on observable behavior and neurobiological measures. For all these reasons, he seems to be an ideal person to have as a keynote speaker for this event, and we're fortunate to have him here. Please welcome Dr. Thomas Insel.
Thank you. That was wonderful. Well, thank you, Rob. That was really a terrific introduction. I wish my family could hear that. And if they ultimately go to CMU, they will, hopefully, at some point. It's great to have such a terrific crowd here. When Subra invited me to do this, I was delighted. I really jumped at the chance. For one reason, I wanted to talk about the Brain Initiative, and I was so excited to hear about BrainHub and to learn more about it. But the other reason, honestly, is that I've begun to realize, which I never knew before, that for people who come to Washington as public servants, if they're really successful, they graduate and go to Pittsburgh. So I was just fascinated to figure out what's the draw, and I'm presuming that you'll have a current president of the United States in Pittsburgh before long. I'm sure you've just paved the way, Subra, for a lot of other great people to come. So it's a delight to be here. I want to tell you rather briefly about this national initiative and to help put in context why I think BrainHub is really an exciting idea, and this is the right time, and this is the right place. So I think we've found that sweet spot that doesn't happen all that often between the world of academics and the world of public policy, and hopefully even the world of financing, which could happen here as well. So the big point to make is that the world has discovered the brain, and as it says on this slide, and as these recent cover articles just from the last 8 months suggest, this is a hot topic. This is what the public has become really intrigued by the brain in all the ways that it might, from understanding our behavior to being able to think about everything from neuromarketing to neuroeconomics to trying to figure out some of the mysteries of why we do what we do. And that has not been lost on publishers, and if you look at books coming out as well, this is the topic of the decade, without question. It's also, as Subra has already said, a public health mandate. Some of that is driven by the demographics, that Alzheimer's will become such an expensive and such a profound public health problem, largely because the largest part of our population, that's in terms of population growth, will be the people over the age of 65. That's just where the numbers are. And Alzheimer's is, after all, and other forms of dementia, these are disorders of late life. You can see here in the study that was done called the Burden of Disease Studies that looks at the public health measure of disability, not just death, but disability measured as something called the DALY, Disability Adjusted Life Year. I'm convinced that probably, Rob, that probably was developed by a very good statistician who loved acronyms as well. But DALYs are one way of trying to capture what is the combination of years lost to disability and mortality. And if you look at that together, you get this single metric. And surprisingly, it's brain disorders that are the leading source of disability, at least in this country, more than heart disease, cancer, arthritis, and lots of other sources that most of us would think about. And the reason that you see these extraordinary numbers for disability is largely because these are the disorders of young people. I mentioned Alzheimer's and dementia as a growing problem, but in fact, when you look at mental disorders, schizophrenia, autism, depression, bipolar disorder, eating disorders, a range of illnesses that are both disabling and often fatal, you see that they begin early in life. That's what that red line is. That's the mental disorders that have a very different life course than cancer or heart disease. In fact, when you talk to adults with a serious mental illness, 75% of the time, they'll report onset before age 25. And that puts these in a very different worldview, and it says that the kind of science that we need to care about, the kind of healthcare we need to think about, is really quite different than the healthcare of late life, which is what so much of our focus has been on recently. So I did mention Alzheimer's, and when you look at late life, you can see this impact in terms of costs. It's, and Subra mentioned already that the very high rate, 50% over age 85, actually it's 1 in 8 over 65 with Alzheimer's. That projection will go from 5 million in the current demographics to something double or triple that by 2050. You can see the costs will go up accordingly. What this graph shows is the opportunity for science to bend that curve. Because if we could simply delay the onset of Alzheimer's by 5 years, we can have a profound impact on the ultimate costs going out into 2030, 2040, 2050. So for us, from the National Institutes of Health, which is really what our mandate is, we look at this next century as the century of chronic non-communicable diseases, much the way that the last century was so much about infectious diseases and diseases of, particularly communicable diseases of children that have been now prevented through vaccines, antibiotics, and sanitation. If we're thinking about chronic non-communicable diseases, often abbreviated by the WHO as NCDs, it's brain disorders that will represent, as I've said, the most disabling and the most costly of these. The problem is that in contrast to the way we think about the heart and the kidney and the lungs and the rest of the body, we really don't know very much about the brain. It's actually stunning. We have the most primitive concepts of how the brain works. And that is perhaps as much as the fact that the brain is now a hot topic and it's popular for the public, but that public health mandate and that profound ignorance is, for me, really the urgent need to begin to change the way we approach studying this most mysterious part of who we are. Now, how do we do that? What's the way in which we can make progress? And there's a big debate going on, but I actually wanted to borrow an insight which I find profound from astrophysics, in this case from Freeman Dyson, who points out that the way that our search for outer space has made progress is through better telescopes and better tools, and that the same thing may be true for inner space. So as he says, new directions in science are launched by new tools much more often than by new concepts. And to skip to the end of this, the effect of a tool-driven revolution is to discover new things that have to be explained. And I think that beautifully describes what the Brain Initiative needs to do. It needs to give us the tools to study the brain so we can begin to actually figure out what are the things that have to be explained. We don't even know that at this point because we have such a gross understanding of how information gets processed in this very complicated computer. We do have some recent progress, and I wanted to take just a few minutes to unpack that because it is so exciting, and it's part of the reason why we're here, because we have this sense that there's now great discoveries to leverage. One set of discoveries have to do with how we can study the anatomy and just the most fundamental structures of the brain. And one approach to this, called Brainbow, has used fluorescent probes that are genetically engineered into, in this case, a mouse brain, to be able to identify all of the enormous ranges of cell types and cell projections. So you can go into a part of the brain in which you can't really separate out individual cells and how they project, and you can stain them in such a way that you can see this. This Brainbow does this in a beautiful way. Josh Sains and Jeff Lichtman have developed this at Harvard. It's been used now for many other purposes. And for the first time, it allows us to disentangle all the elements of complex structures. Going even further into a new kind of neuroanatomy, Carl Deiseroth last year published this technique called clarity. It's a really wonderful story because Carl is a good neuroscientist who developed optogenetics in his laboratory, which has been a profound innovation for understanding brain circuitry. But he invited in a materials scientist, Chung-Young Chung, who had been interested in hydrogels. And Dr. Chung did something that for a materials scientist is actually not that particularly innovative, but transformed neuroanatomy in a way that it really is probably the most important breakthrough of the last 5 decades. And that is, he's taken, in this case, a mouse brain, which you see on the left, and delipidated it. It's taken all the fat out of the brain. The fat is what makes the brain impossible to look through. Replace that with a hydrogel that maintains the integrity of the brain, even at the molecular level. So the brain becomes transparent. That middle panel, the brain is still there. You can't see it until you look at it with a fluorescent scope. And that's what you see in the next panel. The result of that is that it allows you then to create essentially a three-dimensional structure, a three-dimensional brain, but it allows you to sort of fly through the brain and to see the three-dimensional architecture of the brain with all of the cells that are stained and remain intact with all of their projections. This can be stained at a molecular level, so you can see the different molecular components as well. And for the first time, we have, instead of having to sort of put the brain back together by reordering 10,000 sections, you can do this quickly, cheaply, rapidly, in a way that's comprehensive, and you get this image of the brain that we haven't been able to see before. So that's really spectacular for looking at structure. What about function? Well, one of Carl Deiseroth's roommates from college, they both went to Harvard and then both moved to Stanford later, now down the hall from each other, Mark Schnitzer, trained in engineering and then decided to work on aspects of optics. And Mark developed a set of what he calls microscopes. The more official name is pulsed laser microendoscopes, which I think is way overblown. But what these microscopes allow you to do is that rather than taking out tissue and looking at tissue on a slide, these are microscopes that are so tiny they can be inserted into a mouse brain. And they can allow you to look at cellular behavior, cellular activity in real time in animals as they behave. And he's done that here. And again, I have a feeling that I'm going to have the same video problem. I do. So you're not going to be able to see the video, but it's okay. I can walk you through it. The concept is that he uses a dye that lights up the cells whenever the cells are firing. So if the cells are active, they will start to glow. That's what you see on the left. You take a mouse who's in a radial arm maze where there are 8 possibilities where the mouse can run around. And he's asking now a functional question. He's saying, can we decode the signals of the brain? In this case, he's interested in place and space. We know that that is encoded in some odd way in the hippocampus of a mouse or of a human. You have cells for navigation in that part of the brain. So he puts in the fluorescent markers so that the cells, when they fire, it's a calcium indicator dye. Cell will discharge, it'll light up, and then he puts the microscope down, and he can look at thousands of cells simultaneously in the place area of the brain while the animal's running around, and he can ask, what's the code? What is the language of the brain here that we can begin to translate by simply looking at firing patterns? Can we decode it in such a way that we could reverse engineer it? We could actually look at that firing pattern, and even without seeing the middle panel, we could predict where the mouse is in the maze. Not quite there. So he's not able to get the most precise localization. He can tell you which arm of the maze the mouse is on from looking at the code and get it to about 1 mouse length. But he needs some much better computational help and perhaps a few more cells to feed into the model to be able to actually understand the code for place and space. But that's extraordinary, right? That's for the first time being able to read the language of the brain directly, virtually at the speed of thought, in a way that we haven't been able to understand before. I mentioned optogenetics a moment ago, developed by Carl Deiseroth as a way now not only to monitor the pathways and the circuits, but to actually manipulate them. In this case, using channel rhodopsin, a light-activated channel, which is genetically engineered into the part of the brain you care about. It's very specific for just those cells that you want to operate, either up or down. And then by shining light in, you're able to be able to turn on, turn off circuitry with fantastic precision. That has already given us great insights into the relationship of neural circuit activity to very complex behaviors, everything from sleeping to feeding to reward to depression, a whole range of really interesting problems. In fact, a paper that's out just in the last couple of days uses this to essentially help animals recover from a stroke in a way that is very powerful and probably better than anything we've had before. That's great for mice. When you get to humans, the tools that we have are a little less precise, but in some ways no less exciting. So here we have a whole suite of neuroimaging tools that we can begin to put together, looking at structure, function, the temporal relationship of brain areas, and then even doing new generations of molecular imaging. And it's probably going to be from the aggregation of all of these tools that we can begin to get a better handle on how information gets processed, how it gets encoded, stored, and retrieved in the human brain. We're a ways from that. We're nowhere near what we can do with those microendoscopes in the mouse brain. But of course, that begs the question of what it would take to get there. In the case of this particular project, the Human Connectome project, which is being run at many sites, but being led by folks at WashU and University of Minnesota, Mass General Hospital, there have been sort of two sides to it. One has been to try to get better technologies for structure, and this particular image is a way of looking at all of the white matter connectivity, actually the wiring diagram of the human brain at a level that we haven't had before. What it accomplishes, and it's essentially doing this through a computational trick, is it allows us for the first time to disentangle white matter tracks where they cross. The problem for human imaging is we're pretty good at tracking a fiber, and there's some 100,000 miles of fibers in the human brain. We can get a few inches down a particular fiber track until it crosses. The problem in the human brain is things are always crossing. It's a sort of spaghetti city once you get away from the cortex, and that difficulty for us is in disentangling that spaghetti. Well, the Connectome project and the group led by Van Wadeen and Bruce Rosen at Mass General has provided a computational fix for that does allow us, as you can see here, to actually follow fibers through these very complicated areas of crossover. It was with all of these kinds of innovations that the White House decided that the brain would be the next great American project. The president had talked about in his last campaign that if he were reelected, he wanted to have a signature science project for his second term. And there was a lot of debate within the Office of Science and Technology Policy about what that should be, climate versus energy versus shooting asteroids out of the sky. A range of things came up. I think it was partly looking at the recent progress on the brain front in neuroscience, looking at the public health urgency that I already mentioned, that Subra talked to you about, that suggested that this was the way to go. And what I'm particularly excited about is the part that shows up in the green there. If you can read it in the back, it says he wants this to, the Brain Initiative will give scientists the tools they need to get a dynamic picture of the brain in action. So channeling Freeman Dyson and the legacy of what's happened in the search for outer space and astrophysics, this was about tools and creating a next generation of tools. He called for a public and private effort. He said we need to get all hands on deck. He's asked NSF and NIH and DARPA. The FDA has now joined. It's likely the Department of Energy will soon join. IARPA, I think, is going to be part of this. So almost any of the federal agencies that have a stake in either a brain or behavior or computation, to hit your three hub areas, has become part of this. Also, a number of private partners. And you can see some of them here. The Simons Foundation has just announced a global brain initiative. Kavli Foundation and Allen Institute were the sort of founders of much of this through their Brain Activity Map project, which really catalyzed a lot that's happened since. And the Howard Hughes through the Genelia Farm Initiative as a very large supporter of brain science. I don't think I mentioned it here, but I should have, that this is the U.S. Brain Initiative writ large. I'm representing the NIH part along with my colleague Story Landis from the Neurology Institute. But we're one part of a large global effort. There's similar work going on in the EU with an even larger project than what we're doing in the US. China has announced their brain initiative. There's a brain initiative in Israel, which is much more computational. There's a new initiative in Japan. There's one in Australia that will be announced over the next few weeks. And if I were really being complete, I would have included India. Chris, I hadn't even realized until today that India's got a brain initiative as well. So this has become much more than a US effort. It's very much a global effort and a lot of excitement about how to bring all of those together. The NIH piece was captured by this report that was released in June. It was put together by a work group of really leading neuroscientists who brought in lots of engineers and computational scientists and range of other people, including a number of public stakeholders, to help think about what should the NIH Brain Initiative do over the next 10 to 12 years. What should it accomplish? How do you set priorities? How big does it need to be? Really provide us a blueprint. And I'll just take you very briefly through their conclusion. This group was led by Corey Bargman of Rockefeller University and Bill Newsom of Stanford, along with many, many other really brilliant people. They said, go after the circuits, that we have some understanding of the most simple anatomic issues. We have some understanding of behavioral issues and cognitive issues, but we can't bridge them unless we really understand how to map circuits and measure these electrical and chemical patterns within them and figure out how do you cross those boundaries. I yesterday learned the term coarse graining, which I assume many of you know, but it's a way of thinking about how to cross levels of analysis so we can begin to try to explain much more about the way we think and behave in terms of circuit levels of analysis. They gave us 7 areas to pursue. First, to get the parts list through a cell census and cell types, to get at these multiple scales, as I just mentioned, to understand not just structure but function, looking at the brain in action, to go beyond correlational studies, which is so much of what we do today with brain imaging, but to actually think about causality and how to establish causality in going from circuits to behavior, to use models at every level to get to some of the fundamental principles of how the brain works. It's been really interesting to me, and I sort of didn't even realize it until recently, that we always think about the brain in metaphors. We talk about it as a computer or maybe as a muscle that's good for plasticity or as an information processing machine. But virtually all these metaphors probably sell us short because it's likely that there are principles that are latent in brain activity that are not really exemplified in any of the machines that we know about. And part of what this effort would be about in getting these fundamental principles is to get beyond the metaphors, to understand how the brain itself actually works. Human neuroscience has emerged as an important area for us. We've talked a lot about what that might look like. Could we, as you saw in Subra's presentation, could we develop a whole new generation of imaging tools that allow us to do for the human brain what we can do today for the fly brain or for the mouse brain? And then finally, how do we get from all of these initiatives to something of practical value if we really want to begin to understand brain and behavior, both normal and abnormal? How do we get there by putting all this together? The idea was this was about a 10-year, maybe 12-year plan. The first 5 years, they said, follow the Freeman Dyson dictum. You go after the tools. And then in the second 5 years, begin to apply those through much more hypothesis-driven science to fill in the gaps. The budget, they could only estimate and guesstimate about. And of course, living in Washington, we do think a lot about budgets, which is probably why many of us have gotten prematurely gray. But you can see here that their idea was that you spend the money upfront on the neurotechnologies, then you bring in more of the kind of classical neuroscience research and build infrastructure throughout. Altogether, they were projecting something like, we're starting at $40 million a year, so we're actually not as big yet as even the Indian project that Chris has helped to put together, but we'd be growing to something like $500 million a year with a total of investment of about $4.5 billion by 2025. That's a really, in many ways, mirrors what's happened with the Human Genome Project that started out at about $40 million and then grew accordingly in just about this pace and this way. This is not the budget that Congress has agreed to, but it is what these very astute advisors have recommended is required for the task. And it's a tough task. As the president said, it's the next great American project. And if we want to be able to succeed, it's going to require a very significant investment. So to finish up, these are indeed the leading sources of disease burden and cost. That's true already, and it will get even more true with time. We have something to point to. This has been an extraordinary 5 years of technological breakthroughs. For students, there is not a better time to be going into this field. One of my greatest regrets is that I wasn't born 50 years or 40 years later. The things that took me a decade to do, literally a decade, can now be done in a matter of a few weeks. That is just extraordinary. It really is an amazing time in terms of the technology we have. The hope is that we can build on this to create these tools that will really accelerate discovery and build this foundation. And the last thing I want to say, and honestly, part of the reason that I am here, probably more than any other, besides the fact that I'll do whatever Subra asks me to do, but the really key reason is yesterday we had our first council meeting for our brain, it's called a Brain MultiCouncil Working Group. It's 10 different institutes at NIH who are investing in this project to try to make it go in the beginning. And each institute at NIH has an advisory council, and one member from each of those councils comes together for this multi-council working group. And we sat down with them and looked at the first set of grants that have come in, which we're about to announce the awards over the next couple of weeks. But we've, for the first $40 million that we invested in 2014, we had 6 RFAs. We got some 306 applications. We'll be making about 50 some awards. And they looked through that list of awards and they said, where are the engineers? And where are the computational scientists? And where are the material scientists? And we're not seeing enough nanotechnology. You're getting a lot of applications from outstanding neuroscientists. But if you guys want this to succeed, you're going to have to get applications from people you've never heard of. The future of the Brain Initiative is bringing physical sciences and engineering and mathematics, the best computational science we can find, together with neuroscience. That story I told you about Clarity, bringing a material scientist together with a neuroscientist, and in 6 months, they had transformed neuroanatomy more than anybody in the last 5 decades. That's the story we want to tell over and over again. The public health mandate is so urgent. The interest is so urgent. The funding should be forthcoming. This is really the right time to do this, and I really believe this is probably the right place to make that happen. Thanks very much for your attention.